Good morning to all of you. This is our second lecture in this week. Now, we are going to focus a little bit different part. Last time we have been able to see the fermentations. Now this time we'll be going with a respiration. But what sort of respiration? That is bacterial aerobic respiration. Bear in mind, this is aerobic respiration, wherein there will be the participation of oxygen in this particular kind of reactions. So let us understand. Oxygen serves as the terminal electron acceptor for the electron transport chain in aerobic respiration. The aerobic respiration in bacteria typically occurs in three phases: glycolysis, Krebs cycle, and electron transport chain. So oxygen is said to be one of the important part that is associated with all this metabolism part. Basically, glycolysis, we know this is purely an aerobic process wherein there will be oxygen molecules are involved in the process. From glycolysis, we go to Krebs cycle, and from Krebs cycle through electron transport chain, there will be generation of ATP or higher energy compounds in this particular kind of reactions. See here, from glycolysis to pyruvic acid, NADH, and ATP molecule is being generated. Pyruvic acid undergoes or it gets converted to acetyl coenzyme A. It runs into the Krebs cycle where CO2 is released, another ATP molecule is being generated. High energy compounds like NADH and NADH2 are generated, and these are funneled into the electron transport chain, wherein again there will be generation of ATP will be there, oxygen will be there, and ultimately the water molecules are being released in this reaction. So overview of this bacterial aerobic respiration, what it says that it is a process in which there will be generation of ATP molecule with the help of oxygen. See here, terminal oxygen acceptor is there, and therefore there will be water molecules that have been generated. Glycolysis, a very basic part we have already undergone through. I'm not going to go in detail, but a quick review of the same. First stage in carbohydrate metabolism. This is the first stage. From this first step itself, the carbon molecules will be degraded and they will be synthesized or they will be modified into some different forms. So first stage is carbohydrate metabolisms, glycolysis. Oxidation of glucose to pyruvic acid. Oxidation of glucose to pyruvic acid takes place. Located in the cytoplasmic matrix of prokaryotes and eukaryotes. Also called as embedded Mehrab pathway. It does not require oxygen. It occurs when whether oxygen is present or not. So this particular kind of pathway of glycolysis doesn't depend upon oxygen. Okay, so it can happen in non-oxygenic phase as well as oxygenic phase as well. Two molecules of ATPs have been produced that consist of two basic stages. One is the preparatory stage, another is energy conversion stage. See here, you can see this is the preparatory stage, right from glucose to fructose 1,6-bisphosphate. This is preparation phase, wherein there will be utilization of ATP that will take place. And once this is fructose 1,6-bisphosphate is being generated, then it undergoes a reaction wherein there will be generation of pyruvate molecule. This is the energy conservation, energy conserving stage, and there will be ATP that is being released from this particular kind of reactions. Now, alternatives to glycolysis may be pentose phosphate pathway. We have already gone to this one. It is also called as hexose monophosphate pathway or hexose monophosphate shunt. Also, it has been called as. It occurs in cytosol of the cell. No ATP is directly consumed or produced in the cell. Operate either aerobically or anaerobically. Bacteria that utilizes pentose phosphate pathway include Bacillus subtilis, E. coli, Leuconostoc, mesenteroids, and Enterococci fecalis. So we have already gone to this path very quickly. Glucose 6-phosphate and so on and so forth. Generation of glucose 6-phosphate, that is pentose phosphate pathway, has been generated. ED pathway. This pathway also we have seen. The most common route for conversion of hexose to pyruvate, generally found in Pseudomonas, Rhizobium, Azotobacter, Agrobacterium, and Enterobacterium fecalis. See here. From glycerol 3 phosphate to directly pyruvate, ED pathway has been formed or performed. Now, once the molecules of glucosine be degraded, where it does it go? It goes into the Krebs cycle. They also called as the TCA cycle or the citric acid cycle. Large amount of energy stored in acetyl coenzyme A is released step by step oxidative decarboxylation of pyruvate. This is the process that takes place. Pyruvate is converted to acetyl coenzyme A and CO2 molecules are being liberated. It's not a part of Krebs cycle, but it's a majority of acetyl CoA is being generated. NADE gets reduced to NADH. Acetyl coenzyme arises from the catabolism of many lipids, carbohydrates, and amino acids, and ultimately the Krebs cycle has been run. The complete cycle appears by functional in many aerobic bacteria, free living protozoal, most alkane fungi. The facultative anaerobic E. coli does not use the full Krebs cycle under aerobic conditions or when the glucose concentration is high, but does at other times. On Krebs cycles, major function is to provide carbon skeletons for biosynthesis. See this, from glucose, fats, and protein breakdown, acetyl coenzyme is there, and it runs the total Krebs cycle. And once the Krebs cycle has been run, there will be high energy molecules that are being generated, which are being funneled inside the TCA cycle. Correct? So that's all for today. In the next lecture, we'll be concentrating only with the electron transport chain, which is the concluding part of this particular kind of lecture. Thank you for today.